This is your third part of your nervous system set of screencasts, and we are talking about neuron to neuron communication. Now, I do just want to clarify that neurons don't always just communicate with other neurons. Sometimes neurons reach a muscle cell, sometimes they reach another type of cell. It's not always a neuron, but we're going to be referencing when a neuron transmits an impulse between itself and a second neuron. So we just discussed how an action potential is generated. And when we were referencing it, we were talking about the cell membrane of the neuron the entire time, and we were looking at a figure representing the axon. As a reminder, an action or an impulse will travel down the dendrites or in through the dendrites, hit the cell body. This axon happens to be myelinated, so it's going to jump over those myelin sheaths, jumping from node of Ranvier to node of Ranvier. You can see the little Schwann cells illustrated there that make those myelin sheaths. But then it travels all the way to the very end of an axon terminal. And what happens there? Well, if we zoom in at this little spot, we have the axon terminal of what is called the presynaptic neuron. Now the synapse is just the space between the neuron and the next cell that it's communicating with. Neurons do not touch the next structure. So if this was communicating with a muscle cell, wouldn't be touching it. Same thing. This presynaptic neuron, so pre meaning before, synaptic, referencing that synapse, that space, does not touch the dendrite down here of the post meaning after, synaptic neuron. Sometimes you'll see this called like the synaptic cleft. Synapse is just a little bit more simple, so that's why I'm calling it that here. So that impulse has traveled the whole way down. The action potential has reached that reversal of charge, has reached the axon terminal of the presynaptic neuron. What happens? Well, we have to release neurotransmitters from vesicles within the axon terminal, release them into the synapse, where they bind to receptors in the membrane of the dendrite of the next cell. But why and how does this happen? Let's talk about it. So we got structures of the synapse that I just want to identify for you before we go through the steps. Again, we're talking about neuron to neuron communication, not always neuron to neuron though. So presynaptic cell refers to the cell before the synapse that we're looking at. Postsynaptic cell refers to the cell that is after the synapse, that space that we're looking at. So we have this axon terminal of the presynaptic cell. Again, I told you the synapse is sometimes called the synaptic cleft. It's that space between the presynaptic cell and postsynaptic cell. We have the presynaptic membrane, so the cell membrane of that presynaptic cell, the postsynaptic membrane, so the cell membrane of the postsynaptic cell, the neuron. We have some protein channels hanging out in here, and then we have those vesicles called synaptic vesicles. They're those temporary little storage units that you find in cells, and these ones happen to contain neurotransmitters. Neurotransmitters are special chemicals that we'll talk about shortly. And then I also do want to point out to you over here voltage-gated channels. So we talked about sodium voltage-gated channels and potassium voltage-gated channels when we were discussing how an action potential is generated. But these ones that we're talking about here are calcium. So voltage-gated calcium ion channels. That's going to become important shortly. Alrighty. So I think that's what we got. Oh, and to kind of describe this very simplistically, if we're looking at the gated channels down here, the neurotransmitter will bind to them. And that is that particular stimulus that allows that type of channel to open up and allow materials to move in and out of it. And we call that a ligand-gated ion channel. Without the neurotransmitter present, that channel is closed and materials do not move in and out of it. So the neurotransmitter is not actually moving directly in and out of these cells. It's just attaching to these special protein channels found in the membrane of the postsynaptic cell. Alrighty, let's talk about steps of neurotransmission, and we've already reviewed all of these figures. So again, we have an action potential traveling down the axon terminal of the presynaptic cell. Okay, it arrives at the axon terminal. That's the first step. The next step, as a result of this action potential traveling down, that provides the stimulus that those voltage-gated calcium channels need in order to open. When those voltage-gated calcium channels open, that allows calcium to enter the presynaptic neuron. When calcium enters the presynaptic neuron, it's a signal that sends a message to these synaptic vesicles that are carrying the neurotransmitters that it's time to move to the membrane of the axon terminal of that neuron and dock or essentially begin to like merge into that cell membrane. And look at what this process is. We learned about this last unit. Neurotransmitters are released via exocytosis. So we've talked a lot about cell transport as we've learned about neurotransmission. Next up, we have those neurotransmitters enter the synapse and bind to those special ligand-gated channels. Once they bind to those channels, those receptors let materials in that are needed to initiate a new signal in that postsynaptic cell. If the threshold stimulus is reached, great, then we start an action potential and then an impulse travels down that cell. If not, then we haven't reached that all or nothing and an action potential will not be generated in the second cell. Just like I recommended you watch Crash Course Part 2 about action potential, I highly recommend watching Part 3 all about synapse. Or all about the synapse. And as a reminder, bit.lys are case sensitive, so that's 3 lowercase u, 1 capital B, lowercase d, capital R, capital X. At this point, you guys are going to take a brief break and do the neurotransmission cutouts as a practice activity. This kind of walks you through those steps of neurotransmission, what occurs at the synapse. You will have access to printed versions of these. But before you go and do that, if you want to watch the last little bit of the slideshow, that would be great. This is just differentiating between types of neurotransmitters. And maybe this is something that you're going to explore a little bit more in your choice extension activity. But we're just going to differentiate between excitatory neurotransmitters and inhibitory neurotransmitters. I also want to break down this word for you, neuro. Again, we're talking about the nervous system, transmitters. This is the necessary chemical that we need in order to transmit an impulse from one neuron to the next. So that's how I remember what they are. They're just special little chemicals. So an excitatory neurotransmitter will increase the likelihood that a neuron will fire an action potential. Remember how I was just referencing, you need to hit that threshold stimulus. An inhibitory does the opposite. It actually decreases the likelihood that a neuron will fire an action potential. So an excitatory neurotransmitter is more likely to initiate depolarization of the membrane, while inhibitory neurotransmitters are more likely to create a hyperpolarization of the cell membrane, which remember, when a neuron cell membrane is within a state of hyperpolarization, it's unable to generate an action potential temporarily. Glutamate right over here is listed as an example of an excitatory neurotransmitter, and down here we have GABA listed as an example of an inhibitory neurotransmitter. However, not all neurotransmitters are just one or the other. For example, acetylcholine in a skeletal muscle will actually be an excitatory neurotransmitter. However, in a heart muscle, acetylcholine will be an inhibitory, or will act as an inhibitory neurotransmitter. If you go and explore a little bit more about neurotransmitters, you will actually see a bunch that maybe you sound familiar, maybe you've heard of before. So like adrenaline is a specific type of neurotransmitter, dopamine, serotonin, maybe you've heard of releasing endorphins before, and I just referenced acetylcholine as well. I think neurotransmitters are super interesting, and maybe you will too. Last thing I just want to say is it's been such a pleasure to teach you guys this year. It's crazy. This is the last thing that I'll be talking to you about in this bio class, but you're going to learn so much more, and you're going to have such a great sophomore year. And I hope you have a wonderful and healthy summer. And I will see you in the fall. Bye.